Okay, the topic that we'll be discussing now is about the general properties of viruses. Viruses are the most important group in the field of medical microbiology. Now, as we know that the prokaryotic group of microorganisms are the one which are very simplistic in nature and they are unicellular in nature. The examples are being the bacteria, mycoplasma, chlamydia, etc. Some of the eukaryotes such as the lower fungi and protozoa can also be included into the unicellular group of microorganisms. But coming to the viruses, these organisms cannot be taken into consideration as prokaryotes because they have very simple type of morphology and they are not included into the cellular organism. As we can see in case of bacteria, there is a cell wall, there is nucleic acid and certain internal components which are present in the bacteria. But in case of viruses, these organisms are very simple in nature, consisting of only the proteinaceous outer covering and the inner nucleic acid which is infectious in nature. So these are the very simple group of microorganisms without cellular organization. And moreover, these organisms are said to be obligate intracellular parasites because they require a living host for their survival. They cannot survive in the external environment for a longer period of time. They need a host where they can usually attack and get multiplied and carry on their synthesizing the progeny viruses. And hence these are called as obligate intracellular parasite. The reason behind is they do not have the enzyme that helps to carry out their replication or their biochemical cycles external to the host, and hence they completely depend on the host for their replicative processes and synthesis of the progeny, and they use the enzymes of the host to synthesize their progeny virions, and hence these organisms are thought to be the obligate intracellular parasite. And because of this particular characteristic, we cannot grow this organism in the artificial media as we can grow the bacteria in a certain culture media, etc. And hence there is a requirement for the living host to isolate this organism in the laboratory, and these organisms are completely dependent upon the host for carrying out their biochemical cycles. And the multiplication of this organism, that is the replicative process, is very complex in nature. In contrast to the bacteria which usually multiplies by the binary fission, this organism utilizes complex mechanism by which they usually replicate and synthesize their progeny viruses. And moreover, these organisms are not affected by the antibacterial antibiotics, as we can use these antibiotics to inhibit the bacterial group of microorganisms. But here these organisms, because of their simple characteristic feature, they cannot be acted upon by the wide range of antibiotics, and hence we have to go for the interferons or other antiviral agents in order to inhibit the growth of this organism, or in order to inhibit the pathogenesis of this particular group of organisms. And these organisms usually occupy a twilight zone that usually separates the life from the or you can say the living and the unliving group of organism. And it was Stanley in 1935 who was able to crystallize this virus similar to the phenomena of the chemical crystallization, and hence they were demonstrated or they were usually identified as chemical entities. Now after which the extraction of the infectious nucleic acid by Girer and Schramm in 1956 said that this infectious nucleic acid is able to enter into the host and give rise to the progeny virus, and hence these organisms were termed as the chemical entities or the living chemicals, you can say. But later on, with the development of more molecular techniques, we now come to know that these are the smallest living units that we know and are of most important group of microorganisms that helps to understand the chemistry of life. Now coming to the medical importance of this organism, these organisms are mostly involved in the pathogenesis of different diseases, not only causing diseases to the humans but also to the plants and to the bacteria. That is, they have the ability to infect the bacteria, ability to infect the plants, and ability to infect the human host. And these organisms, the diseases that are caused by this organism can range from the mild common flu to the most systemic diseases or the most fatal diseases such as the HIV or the herpes. And they are also the one who are involved in the causation of the diseases that are sporadic in nature, like mumps, endemic like hepatitis, pandemic like influenza, and circumscribed in infections. And moreover, apart from these normal infections, they also have the ability to to cause the tumor infections or the cancers, and these viruses are usually called as the oncogenic viruses. Coming to the morphology of this organism, as we know, they are the simplest group of microorganisms having only proteins and nucleic acid as their components. So coming to the size of this organism, the infectious particle of the organism, that is a virus that is able to induce infection in any host, is called as a virion. So the smallest unit or the progeny virus which is infectious in nature is called as the virion, and they can be distinguished from the bacteria with the help of the filters, that is they have the ability to pass through the filters which can retain the bacteria, and hence these organisms were initially called as the filterable viruses. They have the ability to pass through the filters which can retain the bacteria, and hence the term filterable viruses are usually given to this organism. And now this organism cannot be seen under the light microscope, and hence they can be called as ultra microscopic in nature, but the size of this organism usually varies. The largest virus known is a pox virus measuring about 300 nanometer, which is equivalent to the smallest bacteria, that is the mycoplasma. Now coming to the smallest viruses that we know is a papavirus, which is about 20 nanometers, parvovirus, which is 20 nanometers, and it is equivalent to the largest protein, that is the hemocyanin. Now coming to the method by which we can measure the size of this organism, because these organisms usually can pass through the filters, we can make use of the filters of the known diameter, pore size diameters, that helps us to determine the size of the virus. So the initial technique that was used was called as a rhodocol membrane, which contained colloidal membrane filters of graded porosity. So the organism have the ability to pass through the pore size diameter of this filter, and hence the size can be determined. Later on, many techniques were developed to identify the size of the viruses, the most being the ultracentrifuge, where the rate of sedimentation of the viruses is known and is usually calculated with the help of Stokes law. And next, the recent development in the field of microscopy is the development of electron microscopy, with the help of which we can not only see the size of the organism but also the shape of the organism by using different types of staining techniques. That is, mostly the negative staining is used to identify the shape of this organism under the electron microscope. Coming to the structure and the shape of the microorganisms, the organism, as already described, very simple in nature, having the protein and the nucleic acid. So the nucleic acid is usually enclosed in a capsid which is called as, which is made up of the capsomere. So the external protein which usually protects the nucleic acid is called as a capsid, and hence the whole virion particle is called as a nucleocapsid. Now this capsid is made up of the smallest subunit, that is the capsomere, and it gives a morphology, external morphology to the organism, and not only gives a shape to the organism but also protects the organism from the different types of enzymes such as the nucleases that usually inhibits the nucleic acid. Now when we study of the about the viruses, we should know that it is not the whole virus that is infectious in nature, but it is the nucleic acid of the organism which is infectious in nature. So when we usually degrade the nucleic acid, we make the viruses inactive in nature, and hence most of the hosts which are usually attacked by the viruses produces the nucleases to inhibit the replication of the virus or degrade the nucleic acid so that it is no longer able to cause the infection. And hence the capsid not only protects the organism or gives a shape to the organism but also protects the organism from the enzymes such as nucleases. And the capsomeres are mostly made up of the polypeptides, and they are the one that usually attaches to the host cell and introduces the viral genome into the host cell. In case of bacteriophages, it is we can see that it is not whole virus that enters into the host, but it is the viral nucleic acid that usually enters into the bacterial cell. So apart from giving a shape and protecting from nucleases, this capsid also helps to go and attach to the host cell receptor and introduce the viral genome into the host cell. So coming to the capsid, now what are the different morphology, what are the different shapes of the capsid, and the different viruses usually acquires different shape depending upon the capsid. So the symmetry that is given based upon the capsid is two types. One is called as the icosahedral symmetry and the other one is called as the helical symmetry. Coming to the icosahedral symmetry, it is a polygon symmetry consisting of 12 corners and 20 faces. So it is a polygon. The 12 corners are usually made up of the capsomeres, which are polygon in nature, so and hence they are called as the pentons, and the faces are usually made up of hexons, and hence it is called as the hexagonal capsomere. So in most of the viruses there are 12 pentons, but the number of hexons usually varies depending upon the type of viruses, and the nucleic acid is usually enclosed in this icosahedral symmetry. Coming to the helical symmetry, in the case of helical symmetry we can very well study in the case of tobacco mosaic virus, where the capsomeres and the nucleic acid are wound in the form of a spiral tube. In case of bacteriophages or tobacco mosaic viruses, it is usually rigid in nature, but when coming to the human viruses or the animal viruses, it is mostly pliable in nature. And apart from this, there is a complex morphology also, which is seen in the pox virus. So we have three type of morphologies over here depending upon the capsomeres. One is called as the icosahedral symmetry, the helical symmetry, and a complex symmetry, which is exclusively seen in the pox virus. Coming to the external appendages, in some of the viruses there is an external envelope that is present. So depending upon the presence or absence of the envelope, we can classify the viruses as enveloped viruses and the non-enveloped or the naked viruses. So this envelope is usually derived from the host. After the replication process, after the progeny virion is formed, during the time of release, you can see the envelope being derived from the host cell. So the envelope is mostly made up of lipoproteins. The lipid part of the lipoprotein is usually derived from the host, whereas the protein part is usually synthesized by the viral nucleic acid, or it is viral derived component of the lipoproteinaceous membrane. And the proteins which are usually exclusively synthesized by the virus usually expresses certain types of antigenic substances on their surfaces on the envelope. So these antigenic substances or the peplomers which are expressed on the lipoproteinaceous or the lipid envelopes are usually called as hemagglutinin and neuraminidase in the case of influenza virus. So these peplomers are the one that are antigenic in nature, and towards which the antibodies are usually directed by the host immune responses. So the symmetry of the organism is it has nucleic acid, capsid, and above which some of the viruses can also contain the envelope, and the envelope is usually derived from the host cell, and some of which can be synthesized by the virus, upon which you can see the surface or antigen which are expressed by the organism towards which the antibodies can be directed. So in the case of the influenza virus, there are two types of peplomers or the spikes that can be seen. One is a hemagglutinin spike, which is triangular in nature, and the other one is called as a neuraminidase spike, which is mushroom in nature, both of which are involved in the attachment of the host attachment or to the host and the release of the progeny from the host. Now this envelope or the spikes usually confers three important properties to the viruses. One is a chemical property, the other one is the antigenic property, and the biological property. So and the lipid can be easily solubilized with the lipid solvents such as chloroform, ethers, etc. And the antibodies are usually directed towards the viral antigen, that is the surface antigen, which are the peplomers that are expressed onto the viral surfaces or the viral envelopes. And the biological property of these spikes or the hemagglutinin that can be seen in the influenza is the attachment of the viruses to the host. So we have chemical properties, antigenic properties, and the biological properties. So among the biological properties, it helps in attachment of the viruses to the host. Apart from these hemagglutinin and neuraminidase spikes which are expressed, some of the viruses such as adenovirus also expresses certain types of spikes which are called as fibril spikes. These are also antigenic in nature, exclusively seen in the adenovirus. Now coming to the shape variations, most of the animal viruses are usually spherical in nature. In contrast to the bacterial viruses which have both the type of symmetry. You can see icosahedral symmetry and a helical symmetry. There is a wide variation in the shape when coming to the animal viruses. So most of these animal viruses are spherical in nature. Sometimes they can be irregular, sometimes they can be pleomorphic in nature. And some examples I would like to discuss over here is about the rabies virus, which is usually bullet in shape. We have Ebola virus, which is filamentous in nature, and we have tobacco mosaic virus, which is helical in nature. So apart from this, there can be wide variety of variation that can be seen in the viruses. Coming to the chemical properties, so we know that these organisms are made up of two important components. One is a protein and the other one is a nucleic acid. So apart from that, there are also certain chemicals that can be seen in the viruses. So first we'll discuss in detail about the nucleic acid. There is one nucleic acid that is present. It can be either DNA or the RNA. So protein and the nucleic acid. So nucleic acid is either DNA or the RNA. So this DNA can be either a single stranded DNA or a double stranded DNA. Similarly, coming to the RNA, it can be either a single stranded RNA or a double stranded RNA. And the most important unique features of this organism is that in most of the other group of organism, RNA is involved in the protein synthesis. But here in case of viruses, you can see that this RNA usually act as a genetic material, which is not seen in other group of organism, and hence the nucleic acid is said to be unique in case of viruses. So viruses, in case of viruses, RNA can also act as a genetic material in absence of DNA. So it is only one nucleic acid that can be present, either DNA or RNA. So in the absence of DNA, RNA can act as a nucleic acid, and this can be extracted. The nucleic acid can be extracted with the help of chemical treatment, that is the phenol, and some of the nucleic acid which has been extracted after the chemical treatment still can be infectious in nature, as in case of picorna virus and papavirus, when made to introduce into the resistant group of organism. Usually the nucleic acid or the viruses usually attacks to the host on which they can find the receptor. Specificity is there between the organism, that is a virus, and the host. So when only there is a specificity, it can infect that host and can introduce the viral genome. But in case of this extracted or infectious nucleic acid, without knowing the susceptibility or resistance, you can make the nucleic acid to enter into the host cell and can be infectious in nature. So it is usually a specific character of picorna and the papavirus, where the infectious nucleic acid without the host, that is the capsid, can be made to enter into the resistant group of organism and made to be infectious in nature. And coming to the proteins, the protein apart, that is the capsomeres, as we know, apart from giving morphology to the organism, also expresses certain types of antigens towards which the immune response produces the antibodies and neutralizes the viruses. Next are the lipids. Next important chemical group is a lipid. As we know, it is the only enveloped viruses where you can see the presence of the lipids, and mostly derived from the human host. In case of orthomyxoviruses, it is usually derived during the budding processes or during the release of the progeny viruses. And next group of chemical is the carbohydrates. So only specific group of carbohydrates can be seen in the case of these viruses, apart from which there are certain enzymes. Usually the viruses lack the enzymes, because of which they have to completely depend upon the host, and hence called as the obligate intracellular parasite. But some certain viruses have a specific group of enzymes. In case of influenza virus, this can be very well demonstrated by the presence of neuraminidase. This is the enzyme that helps in the release of the progeny virus. After the infection, the progeny virus is synthesized in the host cell. So during the release process, the neuraminidase enzyme helps in release of the progeny virus. So in the absence of neuraminidase enzyme, the organism cannot be released from the host cell, and hence is made defective in nature. So this organism, in order to get released from the host cell, has to produce an enzyme called as the neuraminidase. So enzymes can also be seen in case of certain viruses such as the influenza virus. And next important group of enzymes that can be seen in certain group of viruses is the transcriptase, that is RNA reverse transcriptase. It is mostly seen in the case of the HIV virus, where the viral genome is usually RNA. So this RNA will get converted to double-stranded DNA and becomes latent in the host, and causes a change in the host cell, which is called as a neoplastia. Now the RNA transcriptase or the reverse transcriptase is exclusively seen in case of the HIV virus and certain group of oncogenic viruses. So chemicals that are present in the viruses are nucleic acid, we have proteins, we have envelopes, that is lipids and carbohydrate and enzymes. Apart from that, we also have a reverse transcriptase. Now coming to the resistance of this organism, these organisms are highly heat labile in nature, which gets degraded or inactivated easily within minutes in 56 degrees centigrade, and within minutes at 36 degrees centigrade, and can be stored for a certain period of time at 4 degrees centigrade. And hence, in order to preserve this organism, we have to decrease the temperature below the freezing point, and can be used and this temperature can be used for a long-term storage. So for storing the various group of organism, we can go for the lyophilization process, or you can go for the freeze drying that usually involves minus 70 degrees centigrade. But one exception over here is a poliovirus, which cannot be stored at very freezing temperature. And coming to the acid resistant or the resistant towards the acid, there are certain group of organisms which are resistant towards the acid, which are the enterovirus, and the acid susceptible viruses are mostly the rhinovirus. And most of the organism gets inhibited in the alkaline condition. Now when exposed to the sunlight, UV radiations or ionizing radiation, they can be easily inactivated. But depending upon the organic matter again in that particular area, which usually inhibits or usually prevents the inhibition of the viruses. Now coming in comparing with the bacterial organism, these are mostly chemical resistance. As we can see, the bacteria are mostly sensitive towards the different types of chemicals. Here these viruses are mostly resistant towards the different type of chemicals. They cannot be easily inhibited by the phenol disinfectants, and the glycerol saline, which is at 50% used to inhibit the bacteria. At this particular percentage, we can store these viruses. So 50% glycerol usually is used to store the viruses in the laboratories, and the presence of molar salts, that is the magnesium chloride and the Na2SO4, this particular chemicals, that is the presence of molar salt in the organic material that is present where the viruses are usually surviving, can protect the viruses from the heat. Next is exposure to the oxidizing agents such as hydrogen peroxide can easily inactivate the viruses, and the effect of chlorination is complete elimination of viruses, again depending upon the organic matter which is present in that particular area. So chlorination can also be ineffective when more amount of organic matter is present protecting the viruses. Coming to the formaldehyde and beta-propionolactone, these chemicals are mostly used to produce the vaccines from the viruses. So exposure of the virus antigen to the formaldehyde or beta-propionolactone can inactivate the virus, but can make this virus as a vaccine, and we can prepare different types of vaccines by exposing the viruses to these particular chemicals. And coming to the lipid solvents, it is usually effective only upon the envelope viruses, and makes ineffective towards the naked or the non-envelope viruses. Antibiotics, here coming to the antibiotics, as already discussed, antibiotics are inhibitory to the microorganism, that is a bacteria, but not to the viruses. Viruses, being simple in nature, cannot be acted upon by the antibiotics. Next important property of the viruses is viral hemagglutination. Now coming to the hemagglutination, it is the agglutination of the RBCs. So most of the viruses have the ability to agglutinate the RBCs at a particular temperature. So these RBCs can be either from the guinea pigs, rabbits, or the humans. So the first demonstration of viral hemagglutination was given by Hurst, who studied this particular phenomena on the influenza virus. So the demonstration of hemagglutinin activity was first in influenza virus by the scientist Hurst in 1941. It is because the virus usually expresses the peplomers which are hemagglutinin in nature, that is are called as the hemagglutinin spikes. And apart from that, there is another important chemical or the spike that is present or expressed on the influenza virus, which is called as the neuraminidase spikes, which helps in release of the viruses from the host. So the presence of this neuraminidase usually destroys the receptors, and hence it is called as the receptor destroying enzymes, so RDE. Now because of the presence of this neuraminidase, it can usually reverse the hemagglutinin activity and helps in release of the progeny viruses from the RBC. So this particular phenomena is called as the elution. Now both this hemagglutinin and elution property can be used in the detection and assay of the viruses, mostly with the orthomyxovirus and the paramyxovirus, who has the ability to express hemagglutinin and neuraminidase spikes. Apart from that, the other detection for this influenza virus is a hemagglutinin titer. It is mostly a quantitative test in order to identify the infectious virions that are present. So in this particular hemagglutination titer, what we do is we have a series of test tubes in which we have the virus isolates that are usually present, to which we'll be adding the RBCs in different suspensions or different dilutions, and the highest dilution or the tube with the highest dilution of the where you can see the hemagglutination is said to be called as the hemagglutination titer. So depending on the hemagglutination titer, you can also detect the infectious virions that are present in a particular solution. And this hemagglutination titer can be carried out either in the test tubes or in the petri dishes, where you can identify the hemagglutinin pattern. If there is no hemagglutinin spike that is present, there is no hemagglutination of the RBCs, which can be identified by the button shape of RBCs, which usually lies like beneath the test tube. So this indicates the negative hemagglutinin assay. Whereas if there is a hemagglutination that is seen in that particular solution, there is a shield-like pattern that can be seen, which indicates the positive hemagglutinin assay. So depending upon the presence or absence of hemagglutinin spikes, you can identify the hemaggglutinating viruses, mostly the myxoviruses. Now apart from that, this hemagglutination titer can also be used to titrate the killed influenza virus, because the ability to cause the agglutination still remains in the killed influenza virus. So you can make use of this hemagglutination titer to titrate even the killed influenza viruses. Now this is about the organism, to detect the organism. Now when we want to detect the antibodies, whether they are produced against this particular organism or not, you can go for the hemagglutination inhibition test. So the presence of antibodies will inhibit the hemagglutination. This indicates that the antibodies specific towards that particular antigen, that is the hemagglutinin, are usually produced in the sera. So hemagglutination inhibition test to identify the antibodies, whereas the hemagglutinin test is to identify the organism. Now apart from that, this test can also be used to purify and concentrate the viruses. And in the case of myxoviruses, there is another pattern, that is the elution that can be seen, where the elution is usually carried out by neuraminidase enzymes or the neuraminidase spike that is expressed onto the host cell. Now one important thing we would like to discuss over here is about the arbovirus. So the arbovirus shows hemagglutinin pattern or the hemagglutinin test positive depending upon the pH and the temperature. So there is a reversal or the agglutination depending upon a specific temperature and the pH. Only at a specific temperature and pH, they carry out the hemagglutinin test positive. Upon the decrease or the increase of the pressure, they usually inhibits the hemagglutination test. So it is in case of arbovirus, you have to keep in mind about the temperature and the pH when you are going to carry out the hemagglutination test. Now coming to the pox virus has the ability to agglutinate the RBCs of the fowls, which can be easily detected with the help of centrifugation test. Next important characteristic feature of this viruses is the viral multiplication. So as already discussed, the multiplication of the virus is very complex when compared to the other prokaryotic group of organism, which usually reproduces by binary fission, budding, etc. But here in case of viral multiplication, as these organisms are obligate intracellular parasites, the replication mechanism of this particular virus completely takes place in the host. Without a host, the organism usually gets inactivated, which cannot survive for a longer period of time in the external environment. And the replication pattern completely depends upon the genetic information which is present in the viruses. That is, as discussed, only one type of nucleic acid can be present. Either it can be a DNA or the RNA. So DNA can be a single stranded DNA or a double stranded DNA, and RNA can be either a single stranded DNA or a double stranded RNA. So based upon that, we will classify the viruses later on. So the best model that was suggested to study the replicative cycle of the viruses was the bacteriophage. So here the similar type of like replication mechanism can be seen even in the animal viruses. So let us classify the different steps of the multiplication. So coming to the phases of viral replication, first is the attachment of the viruses to the host, that is to initiate the pathogenesis, to initiate the infective, or to initiate the replicative cycle of the viruses. First, the virus has to attach to the host cell. So that particular phenomena is called as the adsorption or attachment, followed by penetration, where the viral genome is usually made to penetrate inside the host cell, which is usually infectious in nature. Next is the uncoating. It is mostly seen in case of animal viruses, where the whole virus is usually taken up by the host cell, and uncoating usually takes place by shedding of the external appendages, that is the capsomeres or the capsid, and release of the viral genome into the cytoplasm. Next is the biosynthesis, that is the synthesis of the various viral components, and then we have maturation, where a progeny virion is completely formed, which is infectious in nature. So maturation is the assembly of all the different components into a mature virion, followed by the release of the viruses from the host cell. In case of bacteriophages, it usually causes the lysis of the host cell, whereas in case of animal viruses, most of the animal viruses do not lyse the host cell, but they are released from the host cells. Coming to the replicative cycle in detail, the first step is the adsorption or the attachment. So the adsorption and attachment is a random collision between the viral particle or the virion, which is infectious in nature, with the host cell. Now we should know that there is a specificity that is usually present between the virus and the host cell. Only when there is a specificity, the virus will have the ability to attach to the host cell. Now this specificity is usually determined by the receptors which are expressed on the host cell. It is the reason why most of the animal viruses cannot infect the plant viruses, plants, and the plant viruses cannot infect the animals. So there are certain receptors which are expressed solely by the animal or the animal host cell or the plant host cell, which are expressed onto the host cell and are recognized by the virion particle, and hence thereby the virions usually goes and attach and initiates the infection. So there is a specificity. So the specific receptors has to be expressed onto the host cell. Only then there can be an attachment between the virus particle and the host cell. It is mostly seen in the case of influenza, as already discussed. There are hemagglutination spikes which are present on the host cell, that is which are present on the influenza, which usually recognizes the certain receptors on the host cells. Now as we know that influenza usually is the one that causes the respiratory illness, so the receptor should be present on the cells which are present in the respiratory tract. So here we have the hemagglutination, which is solely present on the viruses, which usually recognizes the glycoprotein receptor present on the upper respiratory tract cells. So there is a specificity. So it is able to infect only the upper respiratory tract and cause the pathogenesis or the infection of the upper respiratory tract or the respiratory diseases. Now next, coming to the HIV virus. HIV virus is the one that usually attacks the immune system of the host and causes the deficiency of the immune system. So the cells that are usually attacked by the HIV are usually the T cells, and the T cell which expresses the CD4 receptor. So here the receptor is a CD4 receptor which is recognized by the HIV virus. So HIV recognizes the CD4 receptors with the help of GP120. So the receptor on the HIV is GP120 is recognized on the T cells which usually expresses the CD4 and hence causes the pathogenicity or the infection of the immune system. Next, coming to the poliovirus. Poliovirus usually recognizes the lipoproteins which are present on the primates. So the one that causes the infection of the primates but not the rodents. Apart from that, as I already discussed, sometimes the nucleic acid that is extracted can also be made to be infectious in nature when given in case of picornavirus, which is usually resistant in case of rodents. Rodents usually do not get infectious with the picornavirus, but when the nucleic acid is extracted, even though there are no receptors on the rodent cells, the infectious nucleic acid can be made to enter into the rodent cell and cause the infection of that particular disease. So there should be a random collision between the receptor and the virus, only then there can be attachment or adsorptions. Next is a penetration, where in case of bacterial viruses, as we know, it is not a whole virion particle that enters into the host cell. The bacteriophage host, that is a ghost, usually attaches to the host cell and it makes the viral genome enter into the host cytoplasm, where it can bring about either lytic cycle or the lysogenic cycle. But in case of animal viruses, it is a bit different, because here in case of animal viruses, the cell wall is not very rigid and hence the whole virus can be engulfed inside the host cell. So the process is similar to that of the phagocytosis, and hence this is usually described as viropexis. So in case of phagocytosis, it is the engulfment of the bacterial group of organism, other group of organism, in order to cause the destruction of the organism. But here in the case of viropexis, it is the engulfment of the virus, where the virus becomes infection and cause the infection of the host cell, or synthesizes its progeny viruses. It is in case of envelope viruses, the envelope is usually taken up in the form of a vacuole, which usually fuses with the cell membrane, and it's taken up inside the cytoplasm, where the next step usually is carried out, which is called as the uncoating. So in the third step, that is the uncoating, there is a stripping of the virus capsid, with the result which gets from it is the release of the nucleic acid into the host cell. So it is stripping of the external part of the viruses, that is a capsid, and release of the viral nucleic acid. It is mostly brought about by the lysosomal enzymes which are produced by the host. So lysosomal enzyme produced by the host usually causes the stripping of the virus to release the viral nucleic acid. In case of the pox virus, there is a slight modification over here. Here we have got two uncoating processes, where the first step is the outer coat of the pox virus is usually removed by the lysosomal enzyme that are synthesized by the host. But in case of the second uncoating, in the cytoplasm, the viral coded enzyme, that is a viral uncoating enzyme, usually uncoats the viruses and cause the release of the nucleic acid into the cytoplasm. And one thing to remember over here is the pox virus usually matures in the cytoplasm. It does not go to the nucleus to get synthesized, but all the processes of the replicative process of the pox virus takes place in the cytoplasm. So hence both the uncoating takes place in the cytoplasm, one which is brought about by the lysosomal enzyme, and second uncoating is by the viral uncoating enzymes. Next is the biosynthesis, very important part of the viruses over here. Now synthesis usually takes place by synthesizing various viral components. Apart from that, synthesizing certain enzyme that has to take over the host cell. Now when the virus attacks the host cell, it, as already discussed, it does not have its own enzyme to carry out its replicative cycle or to synthesize the progeny virus. It has to completely depend upon the host cell to synthesize or to take over the host cell enzyme to synthesize its nucleic acid. So and hence the virus has to synthesize certain protein or the enzyme that usually takes over the host cell metabolism and makes the enzymes to prepare its nucleic acid rather than preparing the other components of the host cell. So the regulatory protein are also synthesized during the synthesis process, which accomplishes the synthesis of different viral components. We have regulatory protein that takes over the control of the host cell, and the site of viral synthesis varies from one viruses to the other viruses. Certain viruses such as DNA viruses usually enters into the host cell nucleuses and synthesizes their nucleic acid and goes to the cytoplasm to synthesize their various components. Exception, as already discussed, pox virus, where all the synthetic machinery takes place in the cytoplasm itself without involving any DNA or the host cell nucleus. Now coming to the viruses which are RNAs in nature, most of the replicative cycle takes place in the cytoplasm. Now coming to the orthomyxo, paramyxo and the retrovirus, Here the synthesis usually takes place in the DNA, and most of the viral proteins are synthesized in the cytoplasm. So in the biosynthetic process, it is the synthesizing the various viral components. So it can be different from different types of viruses, whether they are a DNA virus or the RNA virus. If the DNA virus is there, most of the synthesis takes place, or the viral nucleic acid synthesis takes place in the DNA. If in case of RNA viruses, mostly it takes place in the cytoplasm. But whatever the nucleic acid is there, the viral proteins are mostly synthesized in the cytoplasm. There are variety of steps that usually takes place during the biosynthesis. So when the virus nucleic acid enters into the host, first there is a transcription of mRNA takes place, where later on this transcription of mRNA translates to synthesize the early proteins. These early proteins are also called as non-structural proteins because they are the one that synthesizes various components of the viruses. Next is the replication of viral nucleic acid. Now this viral nucleic acid is very most important component that is involved in the pathogenesis. Later on there is the translation of late or structural proteins. Now here the structural proteins refers to the virion capsid in which the nucleic acid usually gets enclosed. So these are the 4 steps by which the early proteins and the late proteins are synthesized apart from the replication of the viral nucleic acid. Now, as we know that there are different types of genomes that can be seen in the viruses. Based upon that, you can classify the viruses into 6 classes. Class 1 usually comprising about the double-stranded DNA. So, as we know that it is the DNA is a double-stranded one, most of the synthesis of this type of viruses takes place in the host cell nucleus. But in case of hepatnavirus, which is a partially double-stranded DNA, the first complete synthesis of the virus, that is partial DNA which is present in the hepatnavirus, first is synthesized into a complete double-stranded DNA virus in the cytoplasm. Later on it moves to the host cell nucleus where the further replicative cycle usually takes place. But this hepatnavirus when extracted is usually non-infectious in nature, whereas the double-stranded DNAs are mostly infectious in nature. And coming to the pox virus, mostly the pox virions are synthesized in the cytoplasm itself without involving the nucleus of the host, and most of the enzymes are of the pox virus, that is the polymerase of the viruses are mostly involved in the synthesis of the or in the replicative cycle of the pox viruses, where we can see in the other type of organism most of the enzymes of the host are involved in this replicative cycle, but here in case of pox virus it is the polymerase of the virus itself that helps in the replication of the viral nucleic acid but in the cytoplasm. Next is a class 2, which is a single stranded DNA, mostly seen in the parvovirus. So coming to the class 1 example is the adenovirus. Now class 2 is a parvovirus, where the single stranded DNA enters into the nucleus where it is converted to the double stranded DNA. Next is the class 3, which are called as the reoviruses. Here the genome is RNA and it is mostly a double stranded RNA, which usually gets converted to the mRNA, which transcribes the early proteins and the the late proteins and carry out its replicative cycle. Class 4 is the positive sense RNA and the negative sense RNA. So there are 2 types of RNAs over here. One is called as a positive sense RNA and the negative sense RNA. Positive sense RNA is the one that can directly carry out its transcription process. It does not require any other aided help. But in case of negative sense RNA, in example that can be seen in the rhabdovirus, orthovirus or paramyxovirus. Here in the case of negative sense RNA, here the RNA polymerase of the virus itself is used to synthesize or to carry out the replicative cycle, and here the RNAs are mostly not infectious in nature. Coming to the class 6, that is Retroviridae, which is the most important group of microorganisms having single stranded RNA. Best example that we can discuss over here is the HIV virus, and here the single stranded RNA enters into the host nucleus and gets converted to the RNA-DNA hybrid. Now this RNA-DNA hybrid gets converted to the double-stranded DNA. So here from a copy of a single-stranded DNA, we have a double-stranded DNA that usually gets incorporated into the host nucleic acid, causing the virus a latent virus. Now when this latent virus is present in the host cell, it usually modifies the morphology of the host cell and makes it neoplasia, that is expresses new type of antigen on the host host cell. So this is exclusively seen in the HIV, where the reverse transcriptase is the enzyme that helps in conversion of the RNA to the DNA. So first it is the RNA DNA hybrid that is formed. Later on the RNA completely gets converted into the double stranded DNA. Now next important step is the maturation, where we can see the assembly. Now in the biosynthesis process, we have seen about the synthesis of the various components of the viruses. Now all these virus components usually assemble together to form a mature virion. So in the case of maturation, assembly of the various components of the virus takes place to form the progeny virion, which is infectious in nature. So this maturation can either take place in the host cell cytoplasm or the nucleus. So example we'll discuss over here is herpes and adenovirus, which usually matures in the nucleus, whereas picorna and pox virus the maturation usually takes place in the cytoplasm. Now during the release of the virus, there is a budding process which usually encloses a virus in an envelope. So envelope viruses and the non-envelope viruses. So in case of envelope viruses, there is an envelope that has that is usually derived from the host cell. So it is lipoproteinaceous in nature, lipid coming from the host and the protein synthesized by the viruses. So in case of envelope viruses, it can either be from the host nuclear membrane or it can be from the cytoplasmic membrane. In case of herpes virus, the envelope is usually derived from the nuclear membrane, whereas in the case of myxovirus, the envelope usually derived from the cytoplasmic membrane during the budding processes, and after which the organism, the virus expresses the antigens which are in the form of spike. That is, example is the hemagglutination that helps in hematobsorption, that is causing the hemagglutination that helps in detection of the viruses, or which are antigenic in nature. Now the last step, that is the release of the viruses. Once the mature virion is formed, it causes the release of the viruses from the host cell. So the release of the viruses in the bacteria is different from the animal host cell. So in case of bacterial lysis, it completely causes the lysis of the host cell or kills the host cell and gets released into the surrounding medium. But in case of animal viruses, the organism do not kill the cause the lysis of the host cell, but release from the host cell by budding processes or other processes. And here there is a normal replication mechanism going on in the host cell, completely releasing the virion particles. Now once the virion particles are released, they go in hunt for attacking the new cell in which they can cause the infection and again begin the replicative cycle. But in case of the varicella viruses, there is a cell-to-cell contact. That is, the organism cannot remain outside the host cell. So immediately once it gets released from the cell, immediate next cell will be attacked by the organism to begin the replicative cycle. So most of the viruses get released and can remain outside for some time to attack a new cell. But in case of varicella, there is a cell-to-cell contact, and this cell-to-cell contact helps in identifying the cells in the lab diagnosis. Now coming to the polioviruses, here in the case of polioviruses, the polioviruses usually causes much and profuse damage to the host cell while get releasing. So there is a complete declination of the myelin sheath of the when there is a polioviral infections. Apart from that, there is a period that usually takes place during the replicative cycle where we cannot see the presence of the viruses inside the host cell. So the viruses goes underground. This particular phase is called as the eclipse phase, where you cannot see the presence of the virus inside the host cell. So this is called as the eclipse phase. In case of incubation period, that is the time the virus attacks a host cell and gets released from the host cell. In case of bacteriophages, it is only 15 to 30 minutes. But in case of human or the animal virus, it is about 15 to 30 hours. So there is a prolonged period of incubation that can be seen in the animal viruses, and sometimes the virus can also be a latent virus which can cause infection after years together. Now another important topic that should be discussed in the replicative cycle is the abnormal replicative cycles. So this abnormal replicative cycle usually takes place either due to the viruses or because of the cells the virus usually attacks. So there is an incomplete viruses that can be synthesized when there is attack to the cells caused by the influenza virus. So here the hemagglutinin spikes are present, but the organism will not be able to get released out of the host cell. So this particular phenomena is called as a von Magnus phenomena, and the defective is in the phage. The defect is in the phage and not from the host cell. Another important type of abnormal replicative cycle is when the cells, the host cell to which the virus usually attacks. So here the cell will not permit the cell to, the virus to become infective in nature. So these cells are called as the non-permissive cell, and there is an abortive infectious cycle that takes place. And most of the viruses are defective in nature with the defect that they are not able to go and attack the, get released from the host cell and attack the next cell. So this is called as an abortive infection. Apart from that, there are another abnormality replicative cycle where you can see a genetically defective organism. So the best example we'll discuss over here is Rous sarcoma virus. This organism will be able to multiply in the host cell but cannot get released because they are usually enveloped, and the envelope usually is synthesized by the virus nucleic acid, but the organism lacks the mechanism to synthesize its own envelope. So it requires the help of another virus which is called as a helper virus. In case of the helper virus of Rous sarcoma virus is avian leukosis virus. So if the cell is already attacked with avian leukosis virus, this provides the genetic code or the component to synthesize the envelope for the Rous sarcoma virus, where it can become effective in nature. So in the absence of this helper virus, the organism cannot get released because of the lack of the envelope that should be present on the viral progeny. And apart from that, the other defective and helper virus that we can discuss is defective viruses are hepatitis D and adeno associated satellite virus, and the helper virus which helps them in becoming infectious in nature are hepatitis B and adenovirus. Coming to the cultivation of viruses, as we know that these organisms are obligate intracellular parasite, they require a living host for their multiplication, and hence cannot be isolated on an artificial media as we can grow the bacteria. So here we require a living host that can either be an animal inoculation, or you can go with the chick embryo, that is the egg inoculation, or you can go with the cell culture technique, which is recently developed technique for isolation of the viruses. So the most the important step or the initial step by which the virus was isolated was in the animal inoculation. Mostly the human volunteers were used to isolate the viruses and study the characteristic of the viruses inside the human host. The best example was the study of Reed and colleagues, who studied the yellow fever caused by the viruses in the human volunteers. But because there are many drawbacks associated with the human volunteers, that is the most important being the people not coming forward to act as a human volunteer, and moreover immunity can be present in the human host to eliminate the viruses. So these are the drawbacks for which we had to shift the animal inoculation into the experimental animal, mostly the monkeys. So the first isolation of the polio virus was done in the monkeys, which was done by Landsteiner and Popper. Again, because of the risk of handling the monkeys and the immunity problem, again we have to shift to the other group of animals to isolate the viruses. So among which non-human primates were also used to cultivate the viruses, and it was Thielen in 1903 who first used the experimental animal white mice to study the characteristic of the viral infection, and still now this is the this has a wide scope in the virology to cultivate the viruses. And there are certain viruses such as Coxsackie and arbovirus which usually only grows in the infant or the suckling mice. So apart from this particular animal, the organism cannot be isolated elsewhere in the other animal inoculation technique. So exclusively we have to depend upon the infant or suckling mice to study the characteristic of the Coxsackie and the arbovirus and to cultivate these viruses. And depending upon the experimental animal, you have to select the route of inoculation. Either it can be intravenous, subcutaneous, or intraperitoneal, etc. etc. in order to cultivate the viruses. Now the growth can be detected by either the death or the disease or the formation of the lesions depending upon the viral infections, and a serial blind passage has to be carried out to give the confirmatory result for the cultivation of the viruses. The disadvantages associated with the animal inoculation, as already discussed, immunity might interfere and will not allow the organism to grow inside the host. Apart from that, we can use this animal inoculation for the study of the pathogenesis, immune response, epidemiology, and the oncogenesis of the viruses, whether it is oncogenic in nature or not. Next is the use of embryonated eggs. So it has completely changed the field of virology, where it is now very easy to cultivate the viruses in the embryonated egg, which can be easily available and at low cost. It was Good Pasteur who had made use of this cultivation of the viruses in the embryonated egg, which was later on developed by Burnett. So there are wide variety of sites that are present in the embryonated eggs that provides us to inoculate and cultivate different types of viruses. So in the chorioallantoic membrane, we can grow the influenza virus, which can be easily identified with the pock morphology. They form pocks when they grow in this particular area. So development of pocks indicates the presence of this particular organism. So pock counting, each pock is formed by one infectious organism, and hence pock counting helps in identifying the number of infectious particles that are present in the CAM, that is the chorioallantoic membrane. Next we have allantoic cavity, which is used for the cultivation of the influenza, and for the primary isolation of the influenza we can go for the amniotic sac. Apart from that, we have egg yolk sac that helps in cultivation of chlamydia and the rickettsia, which cannot be grown elsewhere in the artificial medium. And coming to the allantoic inoculation, we can make use of this allantoic inoculation to synthesize the vaccines or to produce the vaccine strains that are mostly used in the production of the vaccines. Apart from that, we have duck eggs that can also be used. Mostly the duck's eggs are larger in nature and have more incubation period, can be used to cultivate the rabies virus and to grow the inactivated non-neural rabies vaccine. Coming to the cell culture, so there is a wide variety of variations that are coming forward to cultivate the organisms from the animal inoculation to the cell culture. So the most important being the cell culture, and very easy method to identify and cultivate the viruses in the cell culture. So these cultures, cell cultures technique that are used to grow the viruses was first made use to us by Steinhardt and his colleagues, who was able to successfully cultivate the vaccinia viruses in the rabbit's cornea, that cell culture. And Maitland in 1928 used chalk tissues again to cultivate the vaccinia virus, and the drawback with the cell culture was that it had a bacterial contamination, or there are many possibilities that bacterial contamination can take place in the the cell culture. So in order to reduce the bacterial contamination, we can make use of the antibiotics. As we know that antibiotics cannot be acted upon the viruses, and viruses usually shows resistance towards the antibiotics. They do not get acted upon by the antibiotics, and hence this selective process can be used to inhibit the bacteria, and hence the bacterial contamination can be reduced, and the cell cultures or the tissue cultures can be exclusively used to isolate and cultivate the viruses. And the best you can say the best development in the case of cell culture was given us to by Ender, Weller and Robin, who were able to isolate and cultivate the polio virus in the non-neural origin. Now this organism is highly neotrophic in nature and hence could not be grown in any other organism except the that is, the rodents are usually resistant towards this particular organism, but with the help of cell culture technique, we can cultivate this virus even in the non-neural origin. Now coming to the type of cell cultures, they can be of various types. Cell cultures, they can be of organ culture or explant culture or the cell cultures. Coming to the organ culture, here we can make use of the bits of organs, mostly for the growth of the fastidious group of organisms who exclusively grow grow in that particular organ. Example, we have the tracheal ring organ culture for growing coronavirus. So the coronavirus can exclusively grow in the organ culture which usually has freshly prepared bits of organs. Now coming to the explant culture, they are the fragments of the minced tissues which are usually grown as explants and which are embedded in the plasma clots. This explant culture is exclusively called or selectively called as a tissue culture, in which we can grow different groups of viruses. The best example to cultivate the viruses is the adenoid tissue culture to grow the adenovirus. Now coming to the cell cultures, it is a routine method that is mostly used in the virology to cultivate the different types of viruses. Here there is a dissociation of the tissues to the cell with the help of proteolytic enzyme. Once the tissues have been dissociated into the cells, they are washed, they are counted and grown as a mono layer either in the tubes or the bottles or the petri dishes in the presence of different types of media composition. The media composition in which the cell cultures are usually grown consists of amino acid, vitamins, salts, 5% carbon dioxide, 5% carb, serum, antibiotics and phenol red. So the antibiotics over here is included in order to inhibit the bacterial contamination, and the phenol red is an indicator that provides us indication with the growth of the viruses, like the positive growth or the negative growth depending upon the change in the color of the phenol red. So an indicator has to be added to indicate the growth of the viruses. The media composition has to be accurate in order to give a satisfactory growth of the cell culture. Now these cultures are usually poured or disseminated in the bottles, tubes or petri dishes, where they usually form the mono layers of the sheath on which we usually inoculate with the help of the suspension of the viruses. And some of these modification that are seen in the cell culture is that you can place it in a sloped horizontal position or in the form of a roller drum in order to provide more aerobic condition to grow the fastidious group of organisms. Now the cell cultures can be divided into three types based upon the origin, the chromosomal character and the generations through which it can be maintained. So they are usually classified into three types: primary cell culture, diploid cell culture and the continuous cell culture. So depending upon the primary isolation, you can go for the primary cell culture or the diploid cell culture. Now when you want to maintain it, you can go for the continuous cell lines or the continuous cell culture. Coming to the primary cell culture, here they are prepared from the freshly cultured cell that have been derived from the body, but the drawback of this particular primary cell culture is that it has a limited growth and there is no subculture for it. The most important example of this primary cell cultures are the monkey kidney cell culture, human embryonic cell culture, human amnion, chick embryo cultures, and mostly used for the isolation and cultivation of and vaccine productions. Next is we have diploid cell strains. Here in the case of the diploid cell strain, the original characters of the original diploid chromosome or the karyotypes are usually maintained for serial subcultures, but after growth for about 50 subcultures, the cells usually senesce, and the example of this is a human fibroblast, mostly used for the growth of the fastidious group of organism and also for use for the viral vaccine production. Next is we have the continuous cell lines. Now this is very well derived from the cancer cell. Now the cancer cell has the ability to continuously grow and hence these continuous cell lines are usually derived from the cancer cell. The examples of this are the standard cell lines. You have HeLa cells, Hep2 and Kb cells, and they can be stored in the cold, and hence the maintenance of the organism can be done in the cold storage, and they can be used for the vaccine production. Examples of this is a Vero cell line for the production of the rabies vaccine. Coming to the detection and the virus growth in the cell culture, now we can easily grow the organism in the cell culture, but we should also have the ability to identify the growth of the organism and detect the growth of the organism in the cell culture. So in case of animal inoculation, you can identify either with the death of the organism or the formation of the lesions that are present in the organism depending upon the viral infection. So in the case of this cell culture, there are certain morphological changes that are brought about in the cell culture that help helps in identifying the growth of the organism. So these are called as the cytopathic effects. Cytopathic effects are usually the morphological changes that takes place when the viruses are growing on the cell culture. So this can be studied under the microscope, and the organism that has the ability to produce this cytopathic effect is called as cytopathogenic virus, and the effect is called as the cytopathic effect. So this gives us a presumptive identification for the presence of the virus that are usually growing. So the different cytopathic effect produced by the viruses. So in case of enteroviruses, they usually form the crenation of the cells, and the degeneration of the entire cell sheath can be seen. So this indicates the growth of the enterovirus. Now coming to the measles, they usually form syncytium formation, that is a multinucleate giant cells are usually formed when the organism is growing. Now coming to the herpes virus, they causes the focal degeneration of the cells. Next is the adenovirus, which can be easily recognized by the large granular clumps that are formed, which usually appears the bunches of the grapes appearance. And SV40 organism, the virus can be detected by the cytoplasmic vacuolation. So these are certain cytopathic effects that are brought about by the viruses that helps in easy identification of the viruses. Next is another important characteristic by which we can identify the growth of viruses is the metabolic inhibition. So in a normal cell culture, when there are no viruses, due to the metabolic activity, there is certain acid produced. Now the presence of the acid usually changes the color of the indicator. So in case of the virus which is growing, the metabolic activity is inhibited and there is no change in the color of the indicator that is present. So that helps in identifying the growth of the virus. Next is hematobsorption, mostly exclusively seen in the organism which have hemagglutination spikes. This helps in absorption of the RBCs. So RBCs usually agglutinates, adsorbs onto the cell. So this helps in easy identification of the growth of the viruses. So erythrocytes usually adsorbs onto the cell, which is called as a heme adsorption. Next is another important characteristic of the growth is the interference. So interference is mostly used to identify the non-cytopathogenic viruses. Here the non-cytopathogenic viruses means they are not able to produce any morphological changes. So this can be identified by challenging with the cytopathogenic viruses. Now when the cytopathogenic virus is growing, due to the activity of the non-cytopathogenic viruses, the cytopathogenic activity is inhibited. So this is called as an interference. The activity of non-cytopathogenic virus interferes the activity of the cytopathogenic virus, and hence there is no cytopathic effect that is usually produced. So this is challenged with the cytopathogenic virus, identifying the non-cytopathogenic viruses. So this is called as the interference phenomena. Next is you can go for the transformation, mostly for the oncogenic viruses. These viruses usually forms microtumors where the cells are usually produced giving a pile-up like appearance. So they are called as a microtumor. So identifying the microtumors helps in identifying the oncogenic viruses. Next is the immunofluorescence, which is the very easy method to detect the cells which are usually which harbors the viruses. So here the cells are stained with the help of fluorescent dyes and are observed under the electron microscope or the UV microscope to detect the presence of the viral antigen. Next is the assay of the virus. How do you assay the viruses is either you can go for the total content of the virus, whether they are infectious or not. You have to know just the presence of virus is there or not. Next is to identify the infectious virions. So there are two types of assays that can be carried out to assay the virus or count the number of the virus. So total content of the virus, or that is the total viral particles, or you can go with the infectious virion, whether infectious virions are there and how many infectious virions are there. So coming to the first part, that is to detect the total content of the virus, you can go with the electron microscopic technique by staining them with either simple staining or the negative staining, or mixing them with the known concentration of the latex particle, and the ratio of the latex particles to the viruses can be known to detect the assay of the total viruses that are present in that particular sample. So this is the total viral content. Apart from that, you can go with the hemagglutination titer to detect the presence of hemaggglutinating viruses, that is Coming to the hemagglutination titer, it is easily able to detect the influenza virus or the virus which expresses the hemagglutination spike. So here we have a series of test tube having serial dilutions of the viral virus content, to which the RBCs are usually added. Now the tube that shows the highest dilution which have the hemagglutination like pattern detects the accounts for the hemagglutination titer, and this hemagglutination titer can be carried out only when there are more than 10 to the power 7 infectious influenza viruses. So below that it is difficult to go for the hemagglutination assays, but irrespective of whatever it is the most sensitive method to detect the organism or the virus which expresses the hemagglutinins. Next is the assay of the viruses. The assay of viruses can be classified into two types: quantal assays and the quantitative assays. Quantal assays detects the infectious particles, whether the infectious particles are there or not, whereas in the quantitative assays we detect how many infectious particles are there. So coming to the quantal assays, this can be carried out either in the animals or the eggs or the tissue culture. As we know, depending upon the endpoints, we can identify whether the infectious particles are there or not. So the endpoint can either be the death of the animal or mostly in the case of animal inoculation it is the death. Then you can go for the hemagglutinins or the cytopathogenic effect that are produced in the tissue culture. So these are the endpoints by which you can detect whether the infectious virions are there or not. And it is the titer that is usually accounted over here is the ID50, that is the infectious dose 50, which is calculated with the help of statistical methods using Reed and Munch method. Next is the quantitative infective assays. Quantitative infective assay help us to know how many virion particles are usually present. So initially colony counting, similar to that of the bacterial colony counting, was used to identify the growth of the virus particles. So this colony counting can be done with the help of plague assays. So when the organism is multiplying on a cell, it usually causes the lysis of the cell and a plague is formed. So the presence of the plague indicates the infectious particles. So each plague accounts for as infectious particles. So apart from that, you can go with the POC assay, that is the morphology of the POC can be detected in the chorioallantoic membrane of the chick embryo. Now this was the use of plague assay was usually given us to us by Delbecco in 1952. Here the plaques, the viral suspension to the monolayers of the cell, we add the viral suspensions. After the virus adsorbs to the monolayers of the cell, it is usually replaced onto the solid media. And the plaques are, this is usually done to limit the plaques that are formed. So by limiting the plaques that are formed, it is easy for us to calculate how many plaques are there in that particular area. So it is mostly for the viruses which can attack and infect the other cell and can remain in the external environment for a period of time. But in case of herpes virus, this is not needed because there is a cell-to-cell contact. Once a cell is attacked, immediately next cell will be attacked and there is a limited plaque that is formed. So in case of herpes virus, you can directly count the plaques and easy detection of the infectious particles. Next, in the case of POC morphology, as we know, by growing the virus in the chorioallantoic membrane, these viruses forms the morphological features which are called as the POCs. Each POC is formed by one infectious particle. So counting the number of POCs helps in identifying the number of infectious particles. Next we have oncogenic viruses. The easy method is there is a cell transformation of the microtumors that are formed, indicating the presence of the infectious virions. And vaccinia virus again can be counted with the help of POC morphology. So this is all about the general properties of the viruses.